Hello, my name is Johar, and in this session, I will discuss about galactose, fructose, metabolic pathway, and along with that, I will also discuss about uronic acid pathway and hexose monophosphate shunt pathway. So, although there are a lot of topics, but I'm sure it is a very simple and concise session. Hopefully. And whatever I had not included anything else, I just mentioned only those things that are important as far as exam point of view is concerned. So let's start. Now, first of all, we will discuss about galactose metabolism. So what happens in the galactose after digestion from the there is milk disaccharide, that is the lactose. In the intestine, lactose will be converted into glucose, or you can say precisely broken down into glucose plus galactose by the enzyme lactase. So you already learn about glucose, how it is being metabolized in the glycolytic pathway, glycolytic pathway. Now the rest of the thing is galactose. So here is the galactose, how it will be utilized using the metabolic pathway. First of all, galactose will be converted or phosphorylated into galactose 1-phosphate, and in this galactokinase is the enzyme. That's basically, this is galactokinase. There is some spelling mistake here. Galactokinase. And 1 ATP is used in this reaction. As we can say, ATP will be converted into ADP, and then this galactose 1-phosphate will combine with UDP glucose to make two products. One is the UDP galactose and the other is the glucose 1-phosphate. So UDP galactose will be utilized for the synthesis of lactose, and glucose 1-phosphate has two fate. Either it will be converted back into UDP glucose by combining with UTP, and pyrophosphate is released. It will be converted back into UDP glucose, and again the reaction will proceed while combining with the galactose 1-phosphate. Now this glucose 1-phosphate will be converted into glucose 6-phosphate by the enzyme phosphoglucosidase. I think you have read about this enzyme. This is the second enzyme in the glycolytic pathway. And UDP galactose will be utilized in the synthesis of lactose along with the glucose. It will make glucose, and galactose will make a lactose. You already know that it is a milk. It is present in milk. This is a disaccharide present in milk. Now there may be certain diseases due to metabolic problems. For example, here in case of galactose metabolism, this enzyme, this galactose 1-phosphate uridyl transferase, may be defective, absent, or damaged, or present in low quantity. So in this condition, the disease is known as galactosemia. Galactosemia is an autosomal recessive disorder, and it is difficult for the child to develop normally if this enzyme is absent or defective, because child is mainly dependent on milk initially. However, the treatment is to avoid the milk and milk product, so that there won't be any lactose in the milk, there won't be any lactose in the diet, so there won't be any generation of galactose. So all the ill effects of galactose can be avoided if this enzyme is absent. Milk and milk product has to be avoided in the child. Milk and milk products. This is the only remedy so far. Next is the fructose metabolism. It is perhaps more simpler than the galactose metabolism. Fructose is first of all converted into fructose 1-phosphate, as we have discussed in the galactose. Galactose is converted into galactose 1-phosphate exactly in the same manner. Fructose is converted into fructose 1-phosphate by the enzyme fructokinase, and the enzyme utilizes ATP, and ADP will be released. In the next step, fructose 1-phosphate is broken down into two molecule. One is the glyceraldehyde and the other is the dihydroxyacetone phosphate, and here the aldolase B is catalyzing the enzyme. However, as we learn in the glycolysis, it is aldolase A that catalyzes the reaction that break down the fructose 1,6-bis phosphate into two molecule, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Here if you see, it is only glyceraldehyde, there is no phosphate in there. Here it is dihydroxyacetone phosphate, and we know that dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate, and then glycolytic pathway will continue. So there are two abnormalities in the fructose metabolism. First, the absence of or defect in the enzyme fructokinase. The disease is known as essential fructosuria, and when there is a defect or damage or decreased quantity of this enzyme is present. And the other disease is the deficiency of aldolase B. In this condition, there is a condition known as hereditary fructose intolerance. Normally, whatever the fructose present, it can also be converted into fructose. It can be also phosphorylated by another enzyme, hexokinase. But here, this aldolase B deficiency is more severe, unlike the essential fructosuria. In essential fructosuria, patient usually only excrete excess of fructose in the urine. So essential fructosuria as well as hereditary fructose intolerance, both are autosomal recessive disorders. Now we are coming to the uronic acid pathway. There are many things in the body that are lipid soluble, but if they have to be excreted or metabolized in the body, they first have to be made water soluble by conjugation. Usually it is the UDP glucuronic acid by which things has to be combined first, then making water soluble. Like for example, in the bilirubin conjugation, there will be unconjugated bilirubin is water insoluble, but the conjugated bilirubin is water soluble. So we will learn about why uronic acid pathway is important. So first of all, glucose is converted into glucose 6-phosphate. I think we have all learned about this equation in the glycolytic pathway. This is the first step of glycolytic pathway, and catalyzed by hexokinase. ATP is utilized in this reaction, and then glucose 6-phosphate is isomerized into glucose 1-phosphate. The enzyme is same that we have learned in the last slide, phosphoglucomutase, last slide. And then this glucose 1-phosphate combined with the UTP make UDP glucose, and pyrophosphate is released. The reaction is catalyzed by UDP glucose phosphorylase. Now UDP glucose is again oxidized into UDP glucuronic acid or UDP glucuronate, and in this reaction 2-NADH is released, and UDP glucose dehydrogenase is the enzyme that catalyzes the reaction. So this glucuronic acid or UDP glucuronic acid is used in the conjugation of bilirubin, steroids, and various drugs. There are many drugs that has to be metabolized and excreted after combining with the UDP glucuronic acid. Synthesis of glycosaminoglycans, as you have learned in the chemistry of carbohydrate, there are many glycosaminoglycans present in the extracellular matrix. Now the last is the hexose monophosphate shunt. Some of the equations in these are common in these pathways. Like glucose is first of all converted into glucose 6-phosphate by the same first step of the glycolytic pathway, that is by the hexokinase. ATP is hydrolyzed into ADP, and hexokinase catalyzes this reaction. Next step is the basically it is the oxidation reaction, and in which glucose 6-phosphate is converted into 6-phosphogluconolactone, and the reaction is catalyzed by glucose 6-phosphate dehydrogenase, and NADPH is formed here. This is a reductive biopotential. As we will learn in the fatty acid synthesis, this NADPH is needed. So this reaction is catalyzed by glucose 6-phosphate dehydrogenase, and now this 6-phosphogluconolactone will be converted into 6-phosphogluconate by the enzyme glucuronolactone hydrolase. Now the next step is the 6-phosphogluconate will again oxidize into ribulose 5-phosphate, and there is a decarboxylation at this step also. NADP will be converted into NADPH or reduced into NADPH, and carbon dioxide is released. This reaction is catalyzed by phosphogluconate dehydrogenase. Both the enzyme that is catalyzing the oxidation reaction, glucose 6-phosphate dehydrogenase as well as phosphogluconate dehydrogenase, both needed magnesium ion to function properly. And in both the reaction, NADPH is released or generated. So this NADPH will be utilized in fatty acid synthesis and many other reductive reactions in the body, wherever biosynthetic pathway is going on. In the synthetic pathway, fatty acid synthesis and other synthetic pathway, synthesis and other anabolic pathway. Now there is one problem that can happen because of some abnormality or low quantity of this glucose 6-phosphate dehydrogenase enzyme. It is known as G6PD deficiency or glucose 6-phosphate dehydrogenase deficiency. It is an X-linked recessive disorder, and sometimes in some predisposed individual there are certain drugs that can create a crisis. In this problem there will be hemolytic anemia. Many of the RBC will die because this enzyme generates this NADPH, and NADPH is utilized for the generation of glutathione, and glutathione is an antioxidant. Therefore, because the lack of glutathione, many of the cells, many of the RBCs has got increased oxidative stress, increased reactive oxygen species that lead to damage to the RBCs, and RBC get lysed or die. Due to this death of many of the RBCs, there will be hemolytic anemia. And there is also one fava beans, in which after consumption of the fava beans, because it inhibits this enzyme, therefore people have same kind of symptoms as hemolytic anemia after consumption of this fava beans. That condition is known as favism. So this is all about the hexose monophosphate shunt. We have discussed about the uronic acid pathway, HMP pathway, and fructose and galactose metabolism. So thank you very much.